Guys, so let's start chapter 4 on energy and metabolism. We've mentioned these topics a few times, but now we're going to get into it and understand, where the energy is coming from and how it all works together. So, first of all, we need to talk about how everything runs off of energy. Everything out there, plants, animals, bacteria, all of us need energy in some shape or form. Um, how do we track it? Is bioenergetics that is trying to figure out the flow through the entire system. So we've got the sun, and it is sending out massive amounts of energy, and it is going to get picked up. Those photons are coming down from the sun, and they are going to hit up the photosynthesizers, our plants, our bacteria, all the guys that can photosynthesize, and they are going to be able to harvest the energy from the sun. And then those guys, the producers, are going to be, eaten, go up the food chain, eaten by the consumers. Those guys are going to break down. Everything breaks down eventually, and we can follow the flow of energy through everything. This one, the energy moves to this, moves to this, moves to this, and it goes in a cycle. Oftentimes, when that energy is switching and transferring forms, it can lose. We don't really lose energy, but it can transform into a different form of energy, and that would be heat. So, as we are tracking this cycle of energy through the different organisms and tracking it, that is bioenergetics, tracking the energy flow. Previously, we mentioned in chapter 2, when we talked about organic molecules and putting molecules together and breaking molecules down, we talked about catabolic and anabolic reactions. And so when we subbed all those guys together, those were metabolism. Metabolism, the sum of all the catabolic and the anabolic reactions, endergonic and the exergonic reactions. So just to refresh, here is that pathway one more time. Anabolic is going to take the little bitty guys, and we're going to put the little ones together, and we're going to build them together, and we're going to build a larger molecule when we do that. So we're going to put small things, little bitty small guys, and we're going to bring them together. When we bring them together, we are going to need energy inputted into the system. Energy in, energy comes in. And now we are going to put those guys together, and we've made this large molecule. So that is anabolic. If you want to, how I was taught to remember it was anabolic, and sometimes people use anabolic steroids to bulk up their muscle mass. So if you remember it that way, or however you choose to remember it, anabolic is going to take the small things, and energy comes in, and it will create the bonds to attach them and build a larger substance. That is anabolic. Catabolic is going to take, it's just the opposite. It's going to take the large substance, and it's going to break those bonds. And when it breaks the bonds, it's going to release the energy. So now we have a whole bunch of small things. And when we broke the bonds, energy goes out. Energy is released when you broke those covalent bonds, and we can harvest that energy in the form of ATP. If we were talking about glucose, we would harvest it in ATP. That's going to be our energy currency molecule that we are going to talk about. So again, metabolism is the sum of all of these. Your body is doing this all the time. I just ate a lovely sandwich earlier for my meal, and my body is breaking that down. So right now I am undergoing catabolic. I'm breaking down the food in my system, and I'm breaking down these large particles into smaller particles. And then later I may perform anabolic and use that energy broken down from my food to do work, because I need to do some work around my house. So I am doing catabolic and anabolic stuff all the time. So are you. Everybody is, and that is our metabolism. Examples of these. Catabolic, here is glucose, C6H12O6. We're going to talk about glucose a lot today in this chapter with cellular respiration. Glucose is our favorite molecule for cellular respiration. So here's our glucose, and you, me, all of us that undergo aerobic respiration also need oxygen. And when we have the glucose and the oxygen, we can use these guys, create a reaction, and at the end of cellular respiration, we are going to end up with water and CO2 and energy in the form of ATP. So when we break down the sugars, we end up with energy molecules that we will use later to power things like sodium potassium ATP pumps or a whole bunch of other things in the body. But this is an example of, more time, a catabolic reaction where you take something big and you break the bonds. And when you break the bonds, it's going to release the energy, and that is going to be harvested, transformed into something else. And here's ATP, energy currency of all cells. On the other end of the spectrum, the anabolics. We have the CO2, so our carbon cycle. We're going through this carbon cycle. CO2 is going to be picked up and used by the plants. So plants are going to use CO2. They're also going to pull in some water and combine that together with the light from the sun, those photons coming down from the sun, those energy packets coming from the sun. They are going to be able to combine these things, combine the inorganic CO2, combine the water, and together we are going to build, not we, the plants, the bacteria are going to build glucose and oxygen as their byproduct. So it's, if you notice, it's a perfectly, if we just flip this around, we would have cellular respiration, cell respiration, if we just flip the arrow. So super cool catabolics and anabolic reactions happening all the time. This is happening all the time in plants. Plants are able to photosynthesize, create their own energy molecules when they pull in the CO2, the water, and the energy. They will create the glucose and the oxygen, and then they can use the glucose and the oxygen in their own cellular respiration in the cells throughout their body. So super cool stuff. Catabolism, anabolism, the sum of all of these, both of these, is metabolism. And some of us may have a fast metabolism. Some of us may have a slower metabolism. As you age, your metabolism does slow a little bit as you get older. Because my kids can eat a whole pizza and it is nothing for them because their metabolism is super fast. They are growing like little weeds. Um, our energy carriers, ATP. And then you may see, we are going to mention other molecules. We're going to get to these guys, these electron carrier molecules. We'll talk about them. I don't focus too much about those, especially on Bio 1010, but they do mention them. So I'm going to mention them later on. Our energy currency molecules, NADPs, FADs, those kind of guys. We'll talk about them later. All right, we said that energy is coming from the sun and it's going to go through this cycle with bioenergetics. Energy is going to be defined as the ability to do work to create some kind of change. And when we talk about energy and switching from one thing to the next, we have to remember the laws of thermodynamics. The laws of thermodynamics, the first one is that energy is constant and conserved. We don't just magic energy out of nowhere, it has to come from somewhere. So it is constant and conserved. It's not going to go away, but with our second law of thermodynamics, first law, constant and conserved. Second law is that it can be transferred. The energy can be transferred. So it can be transferred from light energy into chemical energy, like those glucose molecules that we saw that photosynthesis produces at the end. It can totally be transferred. And sometimes when that energy is transferred, some of it, it cannot be gained or lost, but some of that energy will transfer into, be harvested into ATP, and the rest of it will go into heat loss. Is where the rest of that energy may go. Some graphs out there will help you figure out, are we talking about an autotroph or a heterotroph? I like this chart the best when it's talking about how organisms obtain energy because it defines it by energy. This chart over here is also a really good chart. It starts off talking about where the carbon comes from. Is the carbon organic or inorganic? But since this chapter is all about energy, I like this table the best because it asks where the energy is coming from. Is it light or chemical? So we've mentioned light before. If it is light, photo means light. So a phototroph is using light for their energy source. A chemotroph is using chemicals for their energy source. You can guess where we're going to be, but we're going to get there. So we're going to focus back on the light. Phototrophs use light as an energy source. Now, where's the carbon coming from? Is there carbon going to come from CO2, which is inorganic? Or is there carbon coming from organic compounds with those carbon backbones that we talked about in chapter 2? So if it was CO2, we mentioned this one before, CO2 and water combined. These are our photoautotrophs. And examples of photoautotrophs are plants, algae, cyanobacteria. The blue-green algae is what it used to be called, cyanobacteria. Um, Green and purple bacteria can also use it, but they don't have oxygen as their final electron receptor. We won't go into those details, but these guys in general, if they are using light, they are phototrophs. And if they can use CO2 like plants do, they are photoautotrophs. They can make it themselves with the CO2, the inorganic CO2. Organic compounds, photoheterotrophs, they're not using CO2. So maybe they're using something else like glucose or something else out there, some other organic compound. Examples of these are green non-sulfur bacteria or purple non-sulfur bacteria. Let's move on to the non-light user energy sources like us. We obviously cannot just make our food if we stepped out in the sunshine. That would be glorious if we could, but we can't. We are chemotrophs. So we are chemotrophs. Which one are we? Do we use organic compounds or do we use CO2? If something used CO2, it would be a chemoautotroph, just like we saw with photoautotrophs. And there's lots of different bacteria examples of these. I'm not going to list them all, but they are right here. That's why I like this table because it gives you exact examples of different types of energy source animals or bacteria or whatever. So chemotrophs, organic, that's us. That's us. We are chemoheterotrophs because we listed off the four macromolecules that we needed to know, right? In our chapter 2, we needed to know carbs, we needed to know proteins, we needed to know lipids, and we needed to know nucleic acids. Those are organic compounds. They have carbon as their backbone. We are chemoheterotrophs. If oxygen is the final electron receptor, here we are. This is us, animals. And if it was an oxygen, it could be other types of bacteria like Clostridium that can cause tetanus or strep. Those guys don't use oxygen. But this is us over here. We are chemoheterotrophs on this side of the energy table. Speaking of energy, there's two types of energy. There's potential and kinetic. Potential and kinetic. This is a great example. This is potential, right? If this dam happened to break huge volumes of energy, that water would be a force to be reckoned with. Nothing would stand in its way if all of that water got loose from the dam at the same time. So that has potential. The water that is moving, this is kinetic. Same for you and I. If we are moving around, walking from place to place, jogging, taking a hike, going on a bike ride, that is energy of movement. That is kinetic energy. Your cell phone battery, the batteries, any type of battery, that is potential energy. So potential is stored, kinetic is energy of movement. Super easy concepts. We got these. We talked about catabolism and anabolism. We needed energy to come into the system to make bonds and bind things together, or we needed energy. Energy ended up exiting when we broke the bonds, and the energy exited out of the system. So energy in, remember the sunshine was coming in for our photosynthesizers. So this is endergonic right here. And then if we are breaking molecules down and harvesting those broken bond energy in the form of ATP or something else, some other energy currency, but we use ATP. Like this little guy, this guy is a baby chick and it's growing. So I am sure it is undergoing lots and lots of energy that is going to go into this little guy. I am sure it's breaking down lots and lots of glucose to power its growth. So this is an exergonic, and we can see what type of movements are these and energy processes are these. We've got diffusion going across, and then we've got kinetic energy coming down the hill, kinetic energy here. So Inorganic and exergonic. If you had a question talking about energy flow, if energy was being released from a reaction or if energy was going into a reaction, energy releasing from a reaction, exergonic. If energy is coming into a reaction, endergonic. activation energy. We saw that equation for cellular respiration that we're going to talk about in the next video. And it started with glucose, right? It was glucose and CO2. If you had a bowl of sugar sitting on your table and there's oxygen in the environment, right? There's oxygen in our atmosphere. We're not going to turn around and poof, that sugar is going to change instantly into water and CO2. That's not going to happen because you have to activate it. You got to kick off the reaction somehow. So when you kick off a reaction, it is called the activation energy, being able to start that process going. And so there are things out there that can help lower the activation energy, lower the level of energy needed to perform this job. So enzymes are great. Oh, excuse me. Enzymes are great things that can help lower the activation energy needed. So you're still doing the same thing, but it takes less effort to get it started. If I have 3 kids, and somebody's always spilling something. There's always a mess. There's always something to clean up. So if you were cleaning up the spilled Coke or soda or something in the bottom of your fridge, and you have your wet sponge, would it be easier to clean up that stuff with a warm sponge, a warm soapy sponge, or a cold wet sponge? How would what would be easier to get that job done? The warm soapy sponge would be much better at trying to scrub up the dried whatever nasty stuff that is on the bottom of my fridge shelf because somebody spilled some things. That's kind of like the activation energy. What would take less effort? And enzymes help us kick off reactions with less effort, less activation energy. So here it is. In this example, uncatalyzed reaction. And a catalase reaction takes way less energy to do it with catalase than not. Um, so what about these enzymes? How do they work? What do they do? So here's the process of how enzymes work and do what they need to do. With our enzymes, we are going to have, this is our enzyme, and we're going to say for simplicity, if we were in a face-to-face lab, we would do this experiment, and it would be using catalase. Catalase is an enzyme, so I'm going to call this catalase. Catalase enzyme. And on the catalase enzyme, it's a protein, and we said that protein shape matters. We talked about protein came in primary, secondary, tertiary, and quaternary shapes, and that shape was absolutely important. And if it got heated up or denatured in some way, it didn't work the way it needs to. So shape matters on these guys. So here's our catalase enzyme, and this portion here is called the active site. The active site. And if you have catalase, catalase can interact with a substrate. And in this case, the substrate is going to be hydrogen peroxide, H2O2, hydrogen peroxide. And we have catalase in our bodies, so we know what this reaction is going to look like at the end, but let's just walk it through. So peroxide comes along, and it's going to bind at a place called the active site on the enzyme. So our substrate is peroxide, and it's going to bind onto the enzyme at the active site. And when it does, it's called the induced fit because it changes the shape. We had a little triangle shape, but now we've got a kind of prettier semicircle here on the active site. So we've induced fit. The enzyme changes a little bit, so it's not perfectly lock and key. They kind of change and modify each other as they fit in there. Now, we've got the reaction that happens. What happens when you put peroxide on a wound on yourself? If you've ever seen peroxide in action, what happens? We are going to produce products. So the enzyme is going to produce some products. If we're talking about peroxide, our products are water and gas, oxygen gas. We see bubbles, right? If we put peroxide, which is normally just a regular old liquid, and it interacts with catalase, our enzyme, catalase, then it will split apart the peroxide, and we end up with water as a byproduct that stays behind and oxygen that bubbles out of it. So we end up with 2 products at the end if we use peroxide as our substrate and catalase as our enzyme. Our products would be water and oxygen gas bubbles. That's how it works. You have an enzyme, and it's going to have a place on here that's called the active site, and your substrate fits into that active site, and then it will cause a reaction and products come out of the end of the reaction. That's basically how enzymes work. Down. There we go. So sometimes things get in the way. If things get in the way, it's called inhibition. If something is blocking a reaction from happening, it's inhibition. And in this case, we have competitive inhibition. And it is competitive inhibition because here is our active site, and it looks like a half moon, kind of looks like an open Pac-Man mouth or something. And here's our substrate that's trying to bind there, but it can't bind here because this competitive thing came along and stole its spot. It stole the active site. So competitive inhibition happens when something else blocks the active site on the enzyme. So now we can't perform our product, we can't perform our reaction, we can't get the products at the end. This is not going to work with competitive inhibition. But it doesn't always have to be the active site that gets bound. Remember, the shape of the enzyme is absolutely critical to it performing its job. So, what if instead of having our lovely smiley face waiting for our substrate to come along and bind to the active site, something else came along and it bound to a different spot on the enzyme. If it's binding to a different spot on the enzyme, it's called an allosteric site. Allosteric. The allosteric site, it bound with this guy over here. And when it did, it changed the shape. So we're no longer a smiley, half moon shaped thing. We're this weird shape here. Active site no longer works, and we still can't get anything out of this reaction because both of them are blocked. So when both of them are blocked, it's called inhibition. The enzyme's not going to work. It's not going to produce your products if it's under inhibition. That inhibition can be in two types. There's two types of inhibition. There is competitive inhibition, where that substance blocks the actual activation site, the active site, and it can't get in. That's competitive. Competitor basically played musical chairs and stole the spot, and you can't do it. Then non-competitive inhibition is where somebody came along and they're not even chairs anymore. There's no place for you to even sit because somebody came along and bound at the allosteric site. So allosteric inhibition is the same thing as non-competitive inhibition. These are the same terms. Non-competitive inhibition, allosteric inhibition. And inhibition means that the enzyme is blocked at the active site and it cannot attach on and produce some products with that substrate. If we were talking about activation, activation is going to be, here's our allosteric site, here's our activator, and the activator is going to bind into that spot. And now watch, see, now we have this teeny tiny little Pac-Man mouth. And once the activator bound onto it, it changed it. And now the Pac-Man mouth is wide open and it matches the substrate's structure and it works. So in this case, the enzyme was never going to work without an activator. This guy was key. So this is allosteric activation. Allosteric activation. So we have allosteric inhibition that stops the reaction and the enzyme cannot produce its products. And then we have allosteric activation where you wouldn't have had the products without something binding onto the allosteric site. So enzymes, they're pretty finicky and they have to have the exact perfect structure and they have to bind at the active site with their substrate. Just this lock and key induced fit kind of reaction has to happen. Shape absolutely matters and things can affect enzymes. Temperature can affect enzymes. If an enzyme gets too cold or too hot, it could maybe denature, right, and change shape. Different chemicals can affect enzymes. Temperature can affect enzymes. Lots of different things can affect enzymes, and they're super important because remember, they can help reduce our activation energy needed to um, perform a reaction. If we talked about in chapter 2, breaking down milk, cow's milk with its lactose, and lactase is the enzyme that helps you break that stuff down. If you don't have the lactase, remember we said that your gut bacteria has to break it down for you, and it's a different type of breakdown and it makes it uncomfortable. So these are your different terms that you'll need to know what an enzyme is, that it is going to bind to the substrate at a location called an active site. That inhibition means that it's not going to produce any products, and it could be competitive inhibition where it blocks the site and just steals your chair in musical chairs, or it could be allosteric inhibition where there's no longer any chairs because there's nowhere to bind. The site has changed. But inhibition means it blocks it. Activation means that it wouldn't have worked without your activator to come along and change shape. And now our active site is the proper shape for your substrate, and that will then go on to perform and do what you need it to do. Some of these pharmaceuticals, your NSAIDs, these guys can use this methodology, this inhibition or this activation, and this inhibition may come along and it may block your pain receptors. It may get in there and block the site, so you're feeling less pain because of these things. So, enzymes, pharmaceuticals, they can use this um, ability to activate and inhibit different things going on in your body. The last term in this video, feedback inhibition. Feedback inhibition is going to be where you're going to start something. So here we have a substrate, and your substrate is going to bind. We've got Pac-Man, and Pac-Man's going to eat this little triangle at this activation site right here. And reaction happens. Here's our reaction, and we're going to produce a product. Here is our product. And if you see products in a link like this in a cascade kind of action, it's called a substrate. So here's our first intermediate substrate. This substrate is going to bind to a totally different enzyme. And then that reaction is going to produce a totally different substrate. And it will go on and on in this cascade. Now we're down to enzyme number 3, and it's going to produce a different product. And what it turns out is this end product here actually comes along, changes the shape of our first enzyme, this guy up here. We're going to change its shape. And now he's a square shape and he can't bind on. And so what this causes is at the end of this cascade, eventually it shuts itself down. So it's going to have some negative feedback going on. This is feedback inhibition. The product at the end is going to end up turning off the very beginning of this process. That is feedback inhibition, using reactions to regulate the production of something. Feedback inhibition. All right, so that's it for this video. Then the next video, we're going to start talking about cellular respiration.